My name's Dan Mederios. I got my bachelor's degree at the University of Hawaii, Manoa, in zoology, and then I went on to get my PhD at the California Institute of Technology in Pasadena. And I also did postdoctoral work there, became a senior research fellow, which is kind of just a senior postdoc position with some more independence. And then I got my job here at the bio department, the University of Colorado, Boulder, about 2 years ago, and I've been here in my own lab for about 2 years.
Hello. Oh, hey.
Hi, how's it going? We try to understand in this lab how vertebrates evolve from invertebrates, and how can we do that? We're not paleontologists, so we have to use living model organisms, and of course to understand the developmental genetic changes underlying an evolutionary change, you have to use living organisms. So we carefully pick, based on phylogeny, and we pick our model organisms to try to answer this question. So in our lab, we work on 3 main model organisms, but I've been known to work on other species. But the main ones, and the phylogenetically most important ones, are Amphioxus. So Amphioxus is an invertebrate chordate, which means it has a notochord and a dorsal hollow neural tube. Basically has most of the features of a fish, a generic vertebrate, except it lacks, the main thing it lacks is the structures of the head, so it doesn't have a really formed head, so it kind of looks like, in a lot of ways, like a headless fish. So that's Amphioxus is one main model. The other model is our lampreys, so the sea lamprey Petromyzon marinus. It's a jawless vertebrate that has everything almost your generic jawed vertebrate like a fish or a salamander has, except it lacks jaws, so its head skeleton is different. And then we also study zebrafish, which are a really nice, amenable, well-established developmental model system. So to work on Amphioxus embryos, you have to go to Tampa during the breeding season, which is June, July in Tampa, Florida, and dig up the adults and convince them to give you gametes in the lab. Okay, I'll show you the microscope room, which is in here. So it's kept dark because we do fluorescence microscopy, so you need it dark to be able to see. So we have 2 fluorescence scopes here. This is a fluorescence dissecting scope, and so this is kind of the workhorse of the lab. It's typical fluorescence dissecting scope. You can throw your zebrafish embryos, and we have some right here. You inject these zebrafish embryos with a piece of Amphioxus DNA driving expression of a fluorescent protein reporter. You can throw it into the scope, kick on the fluorescent light, and you can visualize where those proteins are being expressed and show you the UV fluorescence. I kind of grew up thinking about these questions. I was one of these kids who would always go out and catch bugs and lizards and put them in jars and keep them. And in Hawaii, it's they always say it's like a natural evolutionary experiment because you have these isolated islands, and you have all these divergent species that are unique to the islands and things. So as a kid growing up, I was always collecting bugs and insects and reptiles, and kind of always thinking about why do things look different from other things, and that kind of just was with me my whole life.